friends, I am Ayantika and in this video I am going to discuss about a brown algae Fucus, and in this video I will be discussing about answer of four questions, that is thallus structure of Fucus species, ecological economic importance of this Fucus, and the reproduction as well as the life cycle. So this will be a entire overview of Fucus species. Overview of Fucus. Now Fucus, it is a brown algae and it belongs to this Phaeophyceae brown algae, which basically means that it will have chlorophyll A and chlorophyll C instead of chlorophyll B. So it has only chlorophyll A and C, and apart from that it has also got carotenes and some accessory pigments like the xanthophylls like lutein, flavoxanthin, violaxanthin, this type of pigments. So it belongs to order Fucales and family Fucaceae. So we are going to discuss about Fucus, and which is also goes by the name of rockweed. And Fucus, in this picture here, the very first one, this one is the Fucus vesiculosus, and the second one is the Fucus serratus, and this the last one is Fucus spiralis. Now it's by habit it is found to be growing in a very intertidal region, shallow region. So by habit it has got modifications for that growing in this intertidal region. For attachment it has got hold fast or hapteron, so that it can stay attached to the substratum. This hold fast or hapteron, they do not penetrate inside the substratum but secrete adhesive liquid, and that helps in attachment with the substratum. So for the attachment with the substratum, it has got hold fast, and just as because it is growing in intertidal region, so during the low tide it will be in outside water. So for that, to protect from desiccation, it is secreting high amount of mucilage. Now mucilage help in preventing desiccation by two process. One is that mucilage absorbs water and it retains water. For the same reason, it is protected. It is a protective material. The mucilage is a protective material that prevents the desiccation of the thallus. So habitat, it is growing in the intertidal region and in cold water. That is why, and also very shallow, as it is growing in intertidal region, we can easily say that it prefers shallow water, and so it is actually in distribution due to this intertidal region and its growth in the shallow water. It is mainly distributed in the northern temperate region of the northern hemisphere. Why only in the northern hemisphere? We can get temperate region also in the southern hemisphere, but because it is growing in a very shallow water, that is in the intertidal region, and we know that in the southern hemisphere the amount of land mass is much less. So that is why it is primarily concentrated in the temperate region of the northern hemisphere, especially in the British Isles and France, coast of France, and also in American continent. You can find it in the Californian coast. So Fucus can be found in this region. Ecological and economic importance. Now this is very common economic importance, is that this is Fucus, it has been traditionally used as a fertilizer. The fucus and along with other brown algae, laminaria, ascophyllum, all these, due to their high mineral content, they are harvested, they are dried and then burned out, so that the ash that is obtained is used as a fertilizer. This is because all these sea weeds or macroalgae of the sea, they accumulate huge amount of mineral nutrient. That is why it is used as a fertilizer. It has been used traditionally also as a fertilizer. So this is one of the most important economic importance. Apart from that, nowadays they are this thalassotherapy, where sea water as well as the weeds of the sea and the mud that is obtained from the sea, all these are put together for improving the healthy condition, for improving the health. So this is thalassotherapy, and many macroalgae, their paste of macroalgae and sea mud, it is used to improve the skin condition. So the fucus is one of the species that is used in this thalassotherapy. Apart from that, it has got much research importance also. Especially this fucus is as a model organism for what study? For the study of cell polarity of cell, and other developmental study, that is studies involving after that is in the zygote, from the zygote to the embryo to the entire gamete, the sporophyte phase. So that entire stage is studied to understand the entire process of cellular development during the embryo development. To understand that process, fucus is used as a model organism, especially for all the macroalgae. Apart from these three economic and research importance, these three, there are also many ecological importance. First is that it is as because it is a massive plant, that is it can grow up to 30 centimeter. So this big you can say it's a macroalgae, big algae. So it is a host for many other small organisms, small other green and blue algae. So this on the surface of the fucus, green and red algae grow epiphytically. So it is a host for growth of these algae, and both red as well as green algae. Thallus structure externally, thallus can be divided into three parts. The lower, here we see the lower region. The lower region, it is a holdfast or hapteron, which is basically a disc-shaped or finger-like projection type of structure, and they do not penetrate the substratum, but they secrete adhesive substance that help it to stick to the surface of the rock on the intertidal region. So the lower part is the holdfast or hapteron. Above it is the stipe region. The stipe region, which is basically the stipe region is basically, it is a cylindrical region. It consists of only the midrib, and above the stipe is very tough, and it prevents the plant to get defoliated from the basal region. So whatever defoliation occur in the upper region, and so the above the stipe, this region above the stipe is a frond, and this frond, it is, as you see, that it has got a central midrib region, this central midrib region, and both sides of the central midrib there is a leaf-like projection which is called the frond, and this frond has got this ear bladders. These are bladders that are filled with air, and at a regular interval these bladders can be found. The main reason why these bladders are found is that they help the plant to buoy to the surface of the water during high tide. During the high tide, the frondage and the branched frondage, they are buoyed to the surface by the help of this ear bladder. Apart from ear bladder, the fronds, that is the leaf-like projection, also has got certain structures, and at the tip, usually the frondage, it gets branched either dichotomously or monopodially. So after the branching, what happens is that at the tip region, which is usually very pointed, which contains the apical cell, the four-sided apical cell. So in the region, after the plant has reached, that is 70% of the height of what the height of a mature plant is. So after reaching that maturity, the tip region, from being very pointed, the tip region, it gets a swelling. Usually, this swollen part contain the sex organs. So the tip region, the apical region, they get swollen due to copious secretion of mucilage, and it contains, on the upper surface, you can see very papillate outgrowth. That papillate outgrowth has a small opening that is called the ostiole. Here we see this. This is the ostiole, and this ostiole, if this is the surface, say for example this is the surface, and you see papillate outgrowth, and this one is the ostiole, and this ostiole leads to a cavity which is called the conceptacle. This is the conceptacle. This cup-shaped structure, it is called the conceptacle, which is borne inside much larger structure called the receptacle. And all this is occurring in the apical region, and in this receptacle, it is borne in the medulla, or the innermost region of the thallus, if we take a transverse section. So in order this entire plant, this is the general feature, that is dichotomous branching or monopodial branching, containing apical region, the sex organs and ear bladders, and the margin is usually, it is very smooth, but it actually depends upon species. For example, if it is Fucus vesiculosus, then it will have a very swollen conceptacle and ear bladder and a very smooth margin. But if we see Fucus serratus, then as the name suggests, serratus, so the margin is serrated. On the other hand, Fucus pyralis will contain, no ear bladder. So and also this Fucus vesiculosus will have a very swollen conceptacle, but if we see the Fucus serratus, it does not have a sex organ concentrated in the receptacle, but it is scattered throughout the apical region. So there is no swelling. So thallus of this thallus of Fucus, it is generally has three parts, but there are minor modification depending upon the type of species. So this is the internal structure. Here we see that the uppermost region, here we see the uppermost region is a single layer of very columnar cells. We see that these are the columnar cells, and each of the cells, they are very tightly packed, and this form a single mono layer of chloroplast containing cells, and each of the single cell of this outer layer, which is called the palisade layer or the meristoderm, that is the palisade layer or the meristoderm, meristoderm. So this each cell has got a large central vacuole, and the central vacuole is surrounded by a thin layer of cytoplasm, and they have got a central nuclei, large nuclei, that is in the vacuolar portion attached with the cytoplasmic fibrils. The cytoplasmic fibril, they keep the nucleus in proper position, and the peripheral cytoplasm, it contains numerous chloroplasts. So this outer layer or palisade layer or the meristoderm, this layer, it has got three function. One is that rapid cell division, another so that there is increase in the surface area and also growth, and another is that second function is that photosynthesis, and the third function is copious secretion of mucilage. So this is the structure of the meristoderm or the palisade layer. The next layer is the cortex. Now we see that this cortex, here this picture, we see the cortex has got polygonal shaped parenchymatous cells, and these parenchymatous cells are multiple layered, and the outer layer cells are usually much larger in size. Here we see these cells are much larger in size, but on the inner side these cells, they are much smaller in size. The outer cell are protective, outer layers are protective, and the inner layer are for storage. And these cortical region cells also, they are vacuolated, but as because this is not the outer layer, outermost layer, so they have fewer chloroplasts, and main function is that the storage. And inner to the cortex, the third layer or the innermost layer, it is the medulla, and we see that medulla has got very long cells. These, so see this long cells, they are called the medullary hypha. The medullary hypha is, the main function of this medullary hypha is that they are placed one next to the other, and they help in the conduction of food matter, and also they secrete copious mucilage, because the sex organs that are born on the conceptacles, which are in turn born on the receptacle area, the receptacle is actually very, it appears swollen due to copious mucilage secretion. The copious secretion of mucilage is from the medulla, and this medulla, in fact the conceptacle lies in the medulla, the receptacle lies in the medulla, and so this main function of this internal region is mucilage secretion, and also that helps in turn protection of the sex organ from desiccation during the low tide region. Reproduction in Fucus. Now Fucus reproduces by two methods. One is the vegetative reproduction, another is the sexual reproduction. So asexual reproduction, that is formation of mitospores like zoospore and the planospore, hypnospore or echinid, all these type of mitospores or this type of reproduction is absent in Fucus. So it has the sexual reproduction is basically oogamous, which is a very advanced feature, and as because it is oogamous, so naturally the sex organs, they are antheridia and oogonia, and as because this Fucus, it is a diploid plant, it is a sporophytic plant, so naturally they will have only haploid stage is the gametes. So gametes are basically the only haploid stage in the entire life cycle. So and the gametes that are the haploid phase, they are produced in this sex organs, that is antheridia and oogonia. But where are they found? What are the position? So these gametes, the sex organs, they are born in conceptacle. So basically at the tip region of the thallus frondage, in this tip region, during the time of sexual maturity, this type of receptacle, this is the receptacle, are found, and in the receptacle we see this cup shaped conceptacle, and this conceptacle open to the outside by the opening called this ostiole, and inside the conceptacle we see these are the reproductive organs. So basically the main idea behind putting this sex organ at the tip is that the tip region, it is reaching the upper layer of the water, and during the high tide and during the low tide they are left exposed, and during the high tide again they get water. So basically for propagation of the gametes, they are placed in the upper region, the top region, and they are protected from desiccation by excess mucilage that is secreted in the receptacle. So basically this is the transverse section of a conceptacle. So this conceptacle, you see that any surface layer of the cell, it acts as a conceptacle initial and starts forming the conceptacle, and the conceptacle has got 2 or 3 layer, this 2 or 3 layer of cells here, these layer of cells, these 3 layer of cells, they form the inner fertile region, from which 2 type of structures develop. One is that from the entire surface, especially this region, this fertile structure, hair-like structure, which can be branched or unbranched, but they are colored. So this colored branched or unbranched hair-like structure are called the paraphyses. Paraphyses, from which the sex organ are developed, or the paraphyses can also remain among the fertile. Some paraphyses, they do not produce sex organ but just remain there among the fertile paraphyses. But at the apical region of the conceptacle, at the region close to the ostiole, here in this region, from this region we see there are also hair-like structure, but they are unbranched and not colored. So these are the sterile branches by the name of the periphyses. Main difference between paraphyses and periphyses are that the paraphyses, that is this structures, this structure that produces this one. This one is a paraphysis that produces antheridium. So this paraphysis is basically very fertile and colored hair-like structure, which can be branched or unbranched, and bears antheridium. But on the other hand, the periphyses, they are unbranched, they are colorless, and they do not produce any sex organ. So basically these are the 3 differences, and there is also difference in the position. Periphyses are born close to the ostiole. Also it can grow over the ostiole, and is a very bushy region that is present in the ostiole. But the paraphyses are present only in the lower part. So these are the 2 type of structures, and the sex organs, that is antheridia, is born on the paraphyses, but oogonia, that is the female organ, they are born from the inner 3 layer of the superficial layer of the inner fertile layer of cells. This one here we see that the antheridia, This one is the antheridia. So basically, this fucus is two types. That is, one is monoecious, where the same plant bears both type of sex organ. So in monoecious species, the sex organs, they are placed either inside the same conceptacle. Male and female both are present inside the same conceptacle. This one is the conceptacle. This one is the conceptacle. So in monoecious species, it can both the sex organ, that is a male and the female sex organ, they can be born inside the same conceptacle, or they can be born in different conceptacle. But in case of dioecious species, in case of dioecious species like Fucus, vesiculosus and serratus, so in this type of dioecious species, sex organ are born in different opposite sex organ, they are born in different plant. That is, there will be a male plant containing the antheridium and the female plant containing the oogonium. So this is how they are placed, and the structures inside the conceptacle. So antheridium, this antheridium is a club-shaped or oval structure that is unicellular and uninuclear at the beginning, and they are born in the fertile hair-like structure called paraphysis. So this fertile filament, which is colored, green-colored filament called paraphysis, bears this antheridium. And this antheridium, it begins as a single nuclear cell, and the single nucleus, it is actually diploid. So undergoes meiosis to produce 4 haploid cells. So these 4 haploid cells, sorry, 4 haploid nucleus, not cell, they undergo further division to form this 23 nuclear stage, and at this 23 nuclear stage, the protoplast undergoes fragmentation or compartmentalization, and after this compartmentalization, actually this 23 cell stage, it is called the, each of the fragment, it is called the sperm mother cell. So this sperm 23 nucleus stage is called this, where each of the nucleus, it is with the surrounding protoplast, is called the sperm mother cell. So gradually this 23 sperm mother cells, by repeated division, they form this 64 sperm cells. These are the diagram, sorry, this one is the diagram where the sperm cells are 64 sperm cells are produced, and each of the sperm cells, they are contained, the entire 64 sperm cells, they are contained in the antheridium, and it has got walls. The outer firm layer is called the exocyte. Sorry, the outer firm layer is the exocyte, and the endocyte, this one, this endocyte or the inner layer is very thin layer. So as the sperm reaches maturity, and especially it reaches maturity when there is low tide, so it absorbs water. The inner endocyte or the inner layer absorbs water and swells, and as because the inner layer or the endocyte, it swells, it creates pressure on the exocyte. So exocyte bursts open, and the endocyte, along with inside containing all the sperms inside it, it is released. And when there is high tide, this packet of sperm is liberated, and it reaches the open sea. during the high tide. So as the sperm packet of sperm with the inside the endocyte, it contains a lot of sperm. So it reaches the open sea. Gradually the endocyte also gelatinizes and releases the sperms. This is the stage where they are released. This one, the stage. So then here how the sperm looks. So we see that the sperm is pyriform or pear shaped, and it has got a two pair of flagella, but it is a heterocont, which means that the two flagella are of different structure. The shorter one, this one here, it is a tinsel type of flagella, which means that flagella will have two rows of femur hair. So the lower, the anterior, that is the forward placed short flagella is tinsel type of flagella, and the long one is the whiplash type of flagella. So the whiplash type of flagella is posteriorly placed, and so the sperm looks pyriform with two flagella, heterocont. Shorter is the tinsel, long and forward directed. Longer is the whiplash type and backward directed, and it has got an eye spot, which is basically it is basically a light receptor, and the sperm moves away from light, and it reacts positively towards gravity. So Ogonium, it arises from the superficial cell, from the three layer of cell that I talked, that lines the inner lining of the conceptacle, the inner lining. This one, inner lining of the conceptacle. See, it contain any superficial cell in this inner lining. It can enlarge, it can gets enlarged and form the ogonial initial. Then the ogonial initial will divide into this this basal cell, this one, this basal cell, and the upper cell. From this upper cell, both the basal and the upper cell do not undergo further division, but the single nucleus of the ogonium, which is diploid in nature, undergoes reduction division to form four nucleus that are haploid. That is, four nucleus will be formed that are haploid, and this four haploid nucleus by further division form the 8 nuclei. That is, four haploid nuclei. Ultimately, they form 8 eggs, or 8. This 8 number of eggs, they are actually they remain together, but they are protected by 3 layer. That is the exocyte, mesocyte, and endocyte. But there is a basic difference between the 3 layer. That is, in the antheridium we see that the exocyte is firm and the endocyte is thin. But here what happens is that in the ogonium, the outer exocyte and the inner endocyte, they are thin. But the middle layer, that is the mesocyte, it is very much hard. So this is the diagram. Here we see that this is the diagram how ogonium it develops, and the exocyte the meso and endocyte, it absorbs water, it swells and creates pressure on the exocyte. So exocyte burst open, releasing the packet of ova, 8 ova, along with the outer mesocyte, the middle mesocyte and endocyte layer. So the egg packet are released with 2 layers. That is the, and later on reaching the during the they are released at this stage, this B stage. It occurs at low tide when they are exposed, and after that when high tide comes, this thing happens. That is the endocyte, mesocyte both absorbs water, but the mesocyte, it gelatinizes. This mesocyte, it gelatinizes it, and the endocyte with the packet of egg cells, they are liberated. So now when the high tide, when the packet of egg cells, they have reached the open sea, so the mesocyte also gelatinizes, only endocyte is left, and later on this endocyte also gelatinizes and the eggs are liberated. Fertilization is external, and as in the previous slide I discussed that egg cell and the sperm, they are liberated when in open sea, and the sperms, they react positively towards gravity. That is, they follow the heavy ova. As it the heavy ova shrinks down, the sperm are also following the ova, and it also shrinks, and the sperms here, they are attracted, the sperms, they are attracted to the ova by a chemical attractant called the fucoceratin. Fucoceratin, this proteinaceous substance, it is a chemical attractant to the that is secreted by the unfertilized egg. So what happens is that after getting attracted by the fucoceratin secreted by the egg, so the sperm, they are attracted and they swim around, and they attaches out of the 2 flagella, one flagella attached to the egg and other flagella keep on lashing. So collective lashing effect of the sperms to the that is attached to the egg, this collective lashing, it causes the sperm, it causes the egg cell to churn out, to swirl in the water. But this entire process, it stops once one of the sperm that remain attached to the egg, one of the sperm, it fertilizes. Now which sperm will fertilize, it will depend upon the maturity, upon the fitness of the sperm. So this sperm fertilizes the egg, and once fertilization is over, the attractant, that is the ferroceratin, its liberation is stopped, and once there is no attractant, so the remaining sperm, it moves away, and the fertilized egg, it secretes a wall around it, that is a zygote, and further germination process starts. So this is the germination process, and once the zygote, it gets encased by a cell wall, it attaches to a substratum, and this type of in the lower region, there is a prolongation, and by then there is a division. The lower cell here, the lower cell, it forms a tubular elongation, and the upper cell undergoes repeated division later to form this club shaped structure here, and we see that the lower cell, it forms the rhizoid. Usually this number of rhizoid increases. Initially the rhizoids are formed, not hapteron, so the number of rhizoid increases, and this club shaped structure gradually converts to this this frond like structure. So this is then ultimate, this H, it is the ultimate stage of germination. Usually the firstlings, it reaches 2 to 3 centimeter, and maximum height that a mature plant after 4 years is generally 30 centimeter. So then the process of germination, its dominant phase is diploid, that is a sporophyte, and so this is a diplontic life cycle where only the haploid phase is in the form of gametes. So meiosis occurs during the gamete formation, that is gametic meiosis. So we begin our journey with the mature thallus of Fucus, which is a sporophyte, and the sporophyte, it forms gametes in the conceptacle at the tip. So undergoes meiosis. This is the oogonium, which forms the eggs, and this is the antheridium, which by reduction division produces the sperm. These are the sperms. So basically this egg is haploid and this sperm is haploid. So the gametes, they fuse to form again the diploid generation. So only this stage is haploid. So the life cycle, this type of life cycle where only zygote gametic meiosis occur, gametic meiosis, so only one stage of gamete, that is haploid. So this type of life cycle is called diplontic life cycle. Fucus is a dominant phase is sporophyte and life cycle is diplontic. So after fertilization we see the young Fucus plant. So this was all about the life cycle, reproduction, thallus structure, both internal as well as external structure, and the economic ecological importance. So do comment on how you like this video, or if you didn't like anything, do also comment so that I can improve. And if you like the video, hit the like button, share it with your friends, and please do subscribe, because your subscription, it encourages me to make more such videos. Thanks for watching this video.